and in today's video, we're gonna go over the adaptive immune response. And I know what you're thinking. You're a veteran Cogni watcher, right? And then you watch all my videos, and you know that I already made a video on the immune response. But this is an even deeper dive into the immune response. That was like level 1. This is level 2, and then maybe tomorrow level 3. My God, this bug in my corner has stayed in my corner for like the whole day. He has not moved. Like, I don't think he's capable of moving anymore. Maybe he's napping. But is he really supposed to nap this long, bro? I don't. This is a question I cannot answer. But anyways, let's keep going. So first off, part 1, the bridge. So transitioning from broad resistance to surgical specificity. So, molecular specificity is the third line of defense, and it relies on lymphocytes, B and T cells, to recognize specific epitopes of a pathogen. This allows the system to distinguish between millions of distinct invaders. Next, we have the immunological memory. And upon initial exposure, the system archives the pathogen profile, and re-exposure triggers a rapid secondary response that usually neutralizes the threat before symptoms appear. And clearly that doesn't work for me because I get sick every single day. And when I get sick, oh ho ho. Whenever I break my hand, it's fine. It's fine. When I get a cold, oh, I'm cooked. I'm just gone. Like no one sees me for the entire week. I'm bedridden. But okay. So we can talk about the messengers, which are dendritic cells. And we have antigen presenting cells, APCs. And these dendritic cells act as crucial links between innate and adaptive immunity, and they phagocytosize pathogens and migrate to the lymph nodes and display digest, digested antigen fragments to the surface to naive T cells. Naive? What? This process effectively sounds the alarm for specific defense systems to activate. See here? It's pretty. I don't know why it's cut off, but not my best graph, not my worst graph. It's not even not my graphs, it's Jerry's graph. How about every mistake Jerry does? Like it's like $10 off his salary. He doesn't make even close to 10. But so we can talk about molecular passwords. MHC ones and twos. I'll give you a second to look at these. It's pretty interesting, I'm not gonna lie. Do, 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 do. Give me your eyes, you're my sunshine. If you know that song, ho ho, you know Peak, my guy. Okay, so antigen presentation requires major histocompatibility complex, MHC. So there's class 1 and class 2. The first one is all nucleated cells, and the function is to provide endogenous antigens for viruses to CD8 plus cells. There's also class 2 for APCs, or dendritic B cells, and they present exogenous antigens to CD4 plus, which are helper cells. Yeah. Next, we can talk about cell 2, which is cell-mediated immunity, and the direct T cell engagement and pathogen elimination. So we have T roles and activation. So helper T. Strike 2. Strike 2. That's strike 2. That's strike 2. Strike 2. So that's $20 off his paycheck for the day. So this activates by MHC2, and they secrete cytokines to coordinate the response, simulating B cells and cytotoxic T cells to multiply. And also, it does cytotoxic T, which activated by MHC1s. And this effector cells target and destroys virtually infected, virally infected or cancerous cells through direct contact. There's also memory T cells, which persist in the blood for decades, and they allow for immediate secondary response if the same epitope is encountered again. Like that's why you get vaccines early on as when you're a child, like hepatitis or like I forgot, like I don't know, I forgot, but you get a lot. Yo, shots are so scary, bro. Like I know it's not gonna hurt that much, but like it's scary. Like I feel like just like you just get some like liquid vaccines. Should I invent that? I'll invent that. I got. Okay, so we're gonna understand the kiss of death. It's very morbid. So mechanisms of killing. So when a cytotoxic T cell recognizes an MHC1 peptide complex, it releases perforons and gramzymes, and this perforinase creates pores in the target cell's membrane, while gramzymes enter to trigger programmed cell death, apoptosis. And this surgical approach ensures that infected cells are destroyed without spilling their viral contents, which prevents further spread to adjacent healthy tissue. As you can see here, you can see an example of this, and I'll let you look at it for a couple minutes so I can scroll on TikTok, you know. Oh, oh, give me your eyes, your mind, sunshine. Why do I even have the calendar app? I don't even look at it. Okay, so now we're gonna talk about part 3, the humoral immunity, the B cell linkage and antibody neutralization. So here's the clonal selection and antibodies. Look at these diagrams. Oh my. You know what? That makes up for Jerry's mistake. So now he only gets minus $20. But here's plasma cells, which are effective B cells that secrete thousands of antibodies per second into the lymph and blood, as you can see here. And then neutralization with antibodies bind to viral surface proteins and prevents them from attaching to host cells. Opsonization, yeah. And then memory B cells, which are long-lived sentinels that provide immediate antibody production upon reinfection. It's pretty interesting. I'll give you another couple of 30 seconds to look at this. I'm kind of tired now. Okay, let's move on. So we have a graph of a primary versus secondary exposure. And as you can see, the secondary response is significantly faster and stronger due to memory cells. And this explains why vaccines provide protection and why you rarely get the same control. Well, clearly not for me. I get a new. If the code is like genetically changing every single day for me, that's happening to me. And if it and my response time is like nothing. But anyways, that's the end of this video. Any questions? Just let me know in the comments and I'll answer them. And a summary is that the adaptive immune system uses molecular passwords and long-term memory to maintain internal homeostasis. It's pretty interesting. Here's image sources. Yeah. Uh, what else can we talk about? I don't know. Who wants to be a doctor? No, I'm not saying like why would someone want to be a doctor. I'm asking like if anyone wants to be a doctor. I thought about becoming a doctor, but then I just thought about how difficult it is, and like how I don't think I do good in high pressure situations. And I don't. I think you need to be. Honestly, your hands need to be steady. Hold on, let me see how steady my hand is. Oh, oh, why am I shaking so much, bro? Yeah, I think this is why I'm not going to medical. But it is impressive when other people do, though. A lot of respect for those people. But anyways, yeah. I hope you learned something cool today, and I will see you in the next video. Bye. Have a good day. See you later. Alligator?